This video outlines the coagulase test, and coagulase is an enzyme that some organisms produce that enable them to clot the host plasma, and plasma is a component of blood. And if they're able to clot this plasma, they actually can create sort of a protective coating around the exterior of the bacterial cell. And it acts a little bit like a capsule in such that it coats and protects the cell and makes it difficult for the host immune cells to phagocytize the bacterium itself. So you can actually work with plasma itself, but we're actually going to use a kit that makes it a bit easier to read. So I've got my starting reagent here, my test strip here, and there's little circles on it. Hold that up so you can take a look at that. And on that you will place the reagent and the bacterium. So the tools that are provided with this particular kit are sort of these kind of hockey stick shaped tools. Because they do have these sharp angles, be cautious when you pick up bacteria not to dig down into the agar. A generic plastic loop would work well as also. So I'm actually going to place the reagent on there, and I really only need one drop, and if you can make it even a small drop, that works well. Okay, because otherwise you have to wait for it to evaporate. So less is more here. And I'm going to place some bacteria onto the drop and mix it in, kind of put it next to it, mix it around. And it takes a few seconds for the reaction to occur. So while I do that, I'm also going to pick up some of the bacterium that I know is going to give me a negative result. Now, the coagulase test is used as a confirmation test, actually, for Staphylococcus aureus. That's the major pathogen that we're looking at here that we're concerned about as having the coagulase activity. So after you've narrowed it down to Staphylococcus aureus, then that would be the time to do the coagulase test at that point as a confirmation test, and that's how it's done clinically. So I have one here that's been sitting a little bit longer, and what I want to show you here is on the left where you see the plus sign. You can see these red clumps that form here. This is a latex particle that's been coated with a protein that will simulate that plasma clumping with the reagent added. And then on the right here, hopefully you can see the negative. So you can see the clumping that's over here on the one side, on the plus, and for the negative you don't see those little specks form. And this organism would be considered coagulase positive, and we would use that as an indicator of pathogenicity. And my other test result has still got a little too much liquid on there, so I don't know if we're going to get a good look at that here. But suffice it to say, once it evaporates a little bit, you'll be able to see that. You can also go back and mix that around a little bit with a plastic loop to see if you see that result here. And I'll do that a little bit more here. I just have a little too much reagent, and you would dispose of those, of course. Yeah, here we go. You would dispose of those in the biohazard. So here we're going to pick that up, and you're starting to see that clumping form under the number 2 circle. And again, after a little bit more time, it'll be a little more distinctive like the one I showed you. But you're starting to see that on the 2 circle form as well. Okay, and that is the coagulase test.